and the bands tend to separate
Right.
from the new.
So you understand the chemical principles and the biological principles of the Meselson-Stahl experiment, and we're going to do that whole discussion in class in a little bit. So that's the perfect sort of question. Hold off on the question, but we'll get there. So the question was, how do we interpret the data figure from the case study? I've got like half an hour blocked out to talk about that. So perfect type of question. I asked you to be able to describe what that figure means. It's a tough figure if you don't understand the basis. So that's what we're here today to talk about. DNA replication. Why do we care? Because this is genetics. And it has the word DNA in it, so you better care. Okay. What does DNA replication allow anything that's a biological organism to do?
Reproduce, grow.
Cell division, cell growth. You are all multicellular organisms, which means you critically depended on DNA replication to be where you are today. Right? All of your cells, with a couple of weird exceptions, have a full copy of your genome, which we discussed last class, the full set of all of the DNA that makes up the chromosomes in both the nucleus and also in the cytoplasm, in your nuclei and in your mitochondria. So you all have this list of vocabulary and tasks. Somehow I jumped ahead and we already covered one of the tasks that I had set for this topic, which was what we talked about last class, predicting the frequency of a given nucleotide sequence in the genome down here. So I've already checked that one off. So really what we're here to do today is modeling. Right? Limitations of models, creating a model, understanding how the model works. This is a little bit abstract, how the chemical basis for the encoding of information by DNA. If you kind of partially understand that, can you tell me what you think that means?
Can you repeat that again?
So that top task, creating a model of the chemical basis for the encoding of information in DNA. If I was going to write that in slightly less wordy language, what should I have written?
Wouldn't it be like if you were given a strand and be able to replicate the second strand?
So being able, okay, so you've got a strand of DNA. How is DNA information? What does DNA have to do with information?
Blueprints for coding enzymes and proteins?
Yes, it's got genes, which are the blueprints that encode enzymes or proteins. So DNA is information. And in fact, I didn't point it out, but in one of the last couple of topics in the course manual, I have, and I never explained this in class, there's a little section of each topic that's called extension material or something like that. Basically, what I did there is if you feel like you want more information about particular topics or especially application to society for some of the topics, those are just resources that you can explore on your own if you're interested. One of those in the previous topic, I think, genomes, was something about Microsoft exploring how to use DNA to store information. I mean, that's what DNA is. You can use DNA just like you can use a hard drive in a computer. You can write, well, I can't. We scientists have the capability of actually synthesizing DNA. We can use the enzymes that synthesize DNA to link together nucleotides in a specific sequence. That's information, just like sentences in languages are information. And so Microsoft apparently is looking into synthesizing DNA that would store data instead of writing it on the hard drive of a laptop. So DNA is information. That's basically the point there. The creating a model part is there's a little exercise later on we'll talk about creating a visual model, a drawing of how information is encoded and passed from one generation to the next by DNA. One thing we didn't talk about to wrap up some of last class's material we looked briefly at sequence alignments. You developed a consensus sequence based on a sequence alignment. This is a little bit different. So hopefully all of you have this in print, digitally, somehow in front of you. We've got 4 different DNA sequences, A, B, C, and D. Can you create a consensus sequence from these? Okay. So if we made a consensus sequence, I'm going to number this alignment. And that's all numbering the alignment, a numbered alignment. When I say create a numbered alignment, it just means every column gets a number so that we can more easily refer to them when we're talking about them. This is something that geneticists do when they look at lots of different copies of, say, the same gene from 4 different species, species A, species B, species C, and species D. So is there a consensus sequence there? Sure, you could make one. What's the consensus nucleotide for the first column? G. Okay, good, everybody's awake. But now what about the second column or the third column?
You would actually have to do 4 different.
Just pick one. There is no consensus in columns 2 or 3. There are 2 C's in column 3. So you could argue with me whether or not you want to say that C is the most common, but sort of getting back to the comment from last class, if you've got 4 sequences and only 2 of them, 50% have C, should we debate about whether or not that means it's more frequent than the others? 50% is only half frequent, which isn't majority, that's just half. What's the point here? Is there a consensus sequence? Are these 4 sequences, in other words, related somehow by sequence? Put another way, these 4 sequences might not be aligned perfectly. So right now, if I sort of sketch this out by making each string of 10 nucleotides align in red, they've all been lined up so that their ends are all flush, so they're even down here. But DNA sequences, when we get them from experiments where we obtain DNA sequences from organisms, those sequences might look something like that up here, where they're not exactly aligned. When we get DNA sequences, it's not all nice and pretty, and the DNA sequencers have started exactly at the same point in determining the gene sequence from one organism and given us exactly the same DNA sequence in terms of its physical location on the chromosome in the other 3. So is there, in other words, any sequence inside that 10 base pairs that makes you or would convince you that those 4 sequences are similar to each other somehow? If you can, which one? ACT. So there's an ACT here, but are there ACTs? So this is what we're looking for. One of the vocabulary words for today is motif. A motif is a small piece of DNA sequence that's, well, between you and me, it's a small piece of DNA sequence that's repeated over and over. It's something you can look for to try to find out if it's present in every sequence you're looking at. So is there a motif?
Is there a length requirement?
Longer is better.
Because I see AC.
So there's a GA in all of them.
AC.
So let's see, there's GA in all of them. Ah, there's a G, well,
Yeah.
hmm.
AC.
So what's going on here?
Just gotta shift the strands around a little bit and they all match up.
Yeah, they all match up eventually. So you found one, which is, there's a GACT there and a GACT there, and there's one here, but we don't see that motif in the 3rd sequence. What else? TA, TA, TA. What else? But more than that.
GCTA on all those first 3.
Okay, is there one in the 4th sequence? No. So the question is, and I'll leave this to you to play with on your own later if you want. The question is, and we're going to do an exercise now with the separate set of DNA sequences to actually do this playing and moving the 4 sequences horizontally with respect to each other. If you rewrote those so that the GCTAs were all lined up with each other, so I'll attempt to show you what I mean in sort of a black dotted line. If you lined up those DNA sequences like that, would you, if you moved the sequences left and right so that those columns were all on top of each other, would you develop something that looks like an aligned DNA sequence? And would that also do something with those that are underlined in blue? So that's how geneticists take raw sequence data, and we don't do this by hand anymore because there's way too much DNA sequence to deal with. We use computer programs that look for motifs and then just move all of the sequences left and right relative to each other to line up vertically motifs. So in other words, which direction would we want to slide horizontally, left to right, sequence D, to line it up with the rest of the sequences? If you grabbed it, you'd have to move it left or right.
Right.
Because you want that blue sequence to be underneath the other 2 blue sequences. So eventually you'd get GACT, T, A, T, A. And what's in between the red motif and the blue motif? A C, and so forth. So this is a bit abstract, doing this without actually looking at moving the letters. So here you go. Maybe a better way to do this. So I'm going to ask you to open a spreadsheet that looks like this in Google Sheets. If you don't have your device with you today, it's going to be a bit tricky to figure this out because you won't be able to actually manipulate the sequence as we go. But it's exactly the same sort of question. So you should all have received a notification in Google Classroom that I just distributed an assignment to you. It's going to have this spreadsheet. So you want to do exactly the same thing. You get about 5 minutes. I'll come around and answer technical questions if I can. It's just me today, no guide on the go from the hub. Same thing though, follow the instruction. Move sets of cells horizontally. Cut and paste might be the best approach to line up those 5 sequences around some sort of a motif. This is not the same set of sequences. This is a new set of sequences unrelated to the one we were just talking about. And if you do have a question, especially technical or otherwise, biological, raise your hand and I'll come around. So actually, what's the motif? Let's settle on that while you're working.
TGC.
TGC. Just a hint. All right. Now I'm going to send you another exercise in Google Classroom. This one's just for fun. Well, they're all just for fun, right? Nervous laughter up in front. Okay, so this is meant to make this point about DNA replication relevant to you. So everyone's going to get a, don't freak out. This is not being scored. This is just for fun. It's not fun, but I'm trying to make it fun. I'm going to show you, as this Google Doc explains, I'm going to show you up here on the monitor a DNA sequence. So what I need you to do is I'd prefer if you have Google Docs open on your laptop or your iPad or your Android device or your Windows device. If you have Google Docs, open it so that you can actually type in letters. Because what I'm going to do is I'm going to show you a DNA sequence, and I want you to copy it down, because you're going to need to use this later. The trick is I'm only going to show it to you for a very short period of time, so I need everybody to be ready to start writing or typing as soon as I put this DNA sequence up here. So you've got 30 seconds to prepare. You can write it on paper if you want to. It's okay if you're not using computer technology, that's okay. No, you can't take a picture of it. No, that's cheating. Actually, it's fine if you want to. Okay. So there's the sequence. Okay. Sounds like people are distracting each other. Okay. So, that was intense.
That was close. That was close.
You were close. Okay, that was intense, yeah. Now, ideally, did anybody do that in Google Docs?
I tried.
You tried. Okay, can you send it to me in Google Classroom, please? Yeah, just submit it, attach it back to the assignment, send it to me. That'll, again, help me get feedback on whether or not people are able to attach things to a Google Classroom assignment. But while you're doing that, what was difficult? You just did DNA replication, by the way. Haha. Sneaky, huh? What was difficult about doing that?
In the English language, we have a thing called space.
So in the English language, we have a thing called spaces. A space is a type of punctuation, right? It helps break up a huge string of characters into things that we, in our minds, can sort of make discrete units. It helps makes it easier for us to remember things. Nobody's memorized that DNA sequence, I assume. But if that had been, probably impossible. That was 100 nucleotides. I give you 100 seconds to copy it. So if you were writing 1 letter per second, which most of you should be able to do, I hope, you should have been able to copy it no problem. So the question is, back to why is it hard to do what I just asked you to do? No punctuation. If I had given you a story with words and spaces, you'd probably be able to tell me that story, mostly. You wouldn't have it exactly memorized, but you'd be able to tell me what was going on. But to us, especially without punctuation, DNA sequence is kind of meaningless. Were there other things that made that difficult?
Yeah, the repetition, double count, sorry.
And also having to convert, so if it's T, you have to convert to A, you don't mind.
So this was true. In real DNA replication, you would have been having to not copy the sequence I wrote up there, but also do the opposite base pair. Instead of a G, a C, and so forth. A's pair with T's, G's pair with C's. I didn't ask you to do that. If you were doing that, that's the idea. That is how DNA replication works. I just wanted you to copy the sequence, but that's okay. That's okay. Now, you also mentioned that part of the difficulty was repetition. So how did that make things harder? Where is there repetition up here? Okay, so there's a bunch of T's, and then I stuttered a bit over here. Right? It's interesting that you point that out because DNA polymerase, which is the enzyme that replicates DNA, another vocabulary word, DNA polymerase, that's the protein that does what you just did. It reads a sequence of DNA and it copies it. More specifically, in your case, it reads one strand and produces the second strand of DNA at the same time. So it's doing both. It's copying, but it's also doing that base pairing at the same time. It has problems with that as well. And that's one of the things we're going to talk about next class is mutations and how they occur. And just temporarily, or just for now, these are particularly problematic for that enzyme because just like you did, it can sort of lose track of where it is when it's copying long stretches of the same letter or when it's copying stutters, like TA, 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 TA. How many times did I do that? 38 or 39? Yeah, whatever. It gives up and it moves on, essentially. Those create mutations, and we're going to talk about that next class. What two needs or forces or characteristics did you have to balance when you did this exercise? Pardon?
Kind of have like chunk it, right?
Yeah, so you,
Kind of count like how many.
so, wait, you're telling me you actually cared whether or not you're doing this accurately?
Yeah.
Okay, so on one hand, DNA replication needs to be accurate because what happens if it doesn't?
Because of the protein doesn't.
Then you have mutations. You've got changes to the DNA sequence, which is not always bad, by the way. Sometimes mutations are good, but the whole point of DNA replication is fidelity. That if you start with one DNA sequence, when you replicate it, you want to have the same DNA sequence. What's the opposing force? You can do it accurately or you can do it time. You can do it accurately or you can do DNA replication quickly. And if you watched one of the pre-lecture videos, you saw my argument for why speed is important. Because if you can't, and this is, by the way, slow. 100 nucleotides in 100 seconds, that's a nucleotide per second of replication. Our enzymes replicate DNA 1,000 times quicker than that. Right? They string together, DNA polymerases, when they're chugging along, replicating our chromosomes, stick together 1,000 nucleotides every second. 1,000, 2,000, 3,000, 4,000, 5,000. That's really fast, and it makes occasional mistakes. So there are DNA polymerases that go slowly and don't make mistakes. There are DNA polymerases that go quickly and do make mistakes.
But doesn't the DNA have an enzyme that proofreads it?
And then our cells have other mechanisms to go back and correct the mistakes. And that's also a topic we'll talk about next class. Next class, again, is mutations and also proofreading. How our cells try to correct mutations or correct errors when they're caught. So we've already touched on the next slides. That was basically the question of why do we care about fidelity or the accuracy of DNA replication. If a mistake gets made, it could be beneficial. There are mutations that help organisms, there are also mutations that hurt organisms. And we're going to talk all about that next class. Mutations and their effects on organisms. To prepare for next class, you might start thinking about your favorite mutation or your favorite mutant. I'm not talking about Teenage Mutant Ninja Turtles either. X-Men, those are always the top two when I do this class, mutations. Free association, people start sending me pictures of Mutant Ninja Turtles and X-Men. But I understand. Okay, so that's sort of the introduction to replication, why it's important and the opposing forces. We need to have replication of DNA act quickly, but also accurately. Now, Meselson and Stahl. There are generally three different models. When Meselson and Stahl did this experiment, there were sort of three different ideas about how DNA might replicate. And I'm just going to focus on two. The third one you do not have to worry about in case you run into it in a textbook or anywhere. The third model is called the distributed model. Don't worry about it. We're focusing on conservative versus semi-conservative replication. So what's conservative replication?
The original strand is intact. Both sides are the original sides of the copy. And there's a completely new strand that's sort of photocopied from the original.
Okay. So, I'm going to try my hand at being an artist. Check it out. That is a photocopy machine. Look, there's the lid. Okay. So the idea of, and this was before, the only information they really had, Meselson and Stahl, this was done decades ago. The only ideas that they had to figure out how DNA might get copied was they knew DNA was a double helix. Watson and Crick and Rosalind Franklin had figured that out before then. So we knew about the double helix. But the conservative model was that you'd stick a single double helix into some sort of an enzyme, and what you'd get out of that was the original double helix, which I'm color coding in black, and a new one. Let's say new strands are in red. That's called conservative because you've conserved the original molecule. The original double-stranded double helix is exactly the same after the process as it was before.
Dr. Ross?
Yeah.
Were they aware of the mRNA or protein synthesis at this point, or were they just, like, if they know about single-stranded DNA and they also know about double-stranded DNA, I don't know, it seems pretty logical to me.
Yep. So yeah, they did know about the existence of both messenger RNAs and proteins at this point in time. But what I'm less clear about is whether or not they understood the relationship between the three when this experiment was done.
I got you.
So how does semi-conservative, which is the way DNA replication actually happens, semi-conservative, not what I just drew up here, how does that work?
Each of the parental strands is part of the new strand.
Right. So that's why it's semi or half conservative. You start with that double strand, and instead of going through a photocopy machine, you denature or melt the strands. We've talked about that concept before. You break the hydrogen bonds that hold the two strands together by what process normally? What we've talked about before anyway was heat. So if you heat a DNA molecule, those hydrogen bonds break and the DNA becomes single-stranded. DNA replication does not involve heat. But you make those molecules single-stranded so that their nucleotides are sticking out kind of like broken rungs on a ladder here. And then, what's the essence of semi-conservative DNA replication?
Half a strand is conserved.
So, why is it useful that we have half a strand over here and half a strand over there? What is a half a strand? That's information. It provides the basis for the synthesis of the opposite strand using base pairing rules. right? So if I draw in an actual DNA sequence, AGTC, let's see, what was the other black strand, top to bottom? So originally, that black strand was the original parental DNA molecule. We separated it. So now each of those strands is the template or the parent strand for the synthesis of a daughter strand. So in that case, the red strand on the left is T, C, A, G by base pairing. And I'll leave it to you to fill in the other newly synthesized or red strand. Look, it's the same as the black strand on the far left. And you get two identical DNA molecules. They're identical in sequence. So that's the concept of a template strand and a daughter strand, or a newly synthesized strand, whatever you want to call it. It's the new strand of DNA. Any questions about that? So that's semi-conservative, half conservative. Half of the strands are old, half of them are new.
Is the denaturing mechanism hydrolysis here, or?
So in this case, in the case of DNA synthesis, which we're going to see again in a minute, but I will certainly answer now, we have an enzyme, a protein, that is basically like a wedge. It shoves itself in between the strands and pulls them apart. It's called helicase. Preview of things to come in like 2 slides. But yes, helicase is the name of the enzyme that separates the 2 strands. Here we go. All right, so semi-conservative is here down below. That's what actually happens during cell division. Questions? So Meselson and Stahl's experiment is all about figuring out which of these 2 models actually happens. And we've already got the answer. We know it's semi-conservative. The point of reading the manuscript was understanding how their experiment works, because on the exam, I'll be asking you to apply the principles of the Meselson-Stahl experiment in a new way that we haven't seen before in class. So it's important to understand how this works so that you can use it, apply this knowledge later. So let me set up the situation here. We're going to start with a population of 4 bacteria. Meselson and Stahl worked with E. coli. And what do you know about E. coli? About their DNA?
It's plasmid.
Yep. Bacteria have plasmid DNA, which is circular. So these things that I'm drawing in black, it's very important to distinguish the circles from the circles. So the black circles are the bacteria. And I'm going to draw inside them, or at least I'm going to attempt to draw inside them, their circular genomes. So what I'm setting up here is 2 rounds of cell division. Four cells become 8, 8 cells become 16. Everybody okay so far? Exponential growth. Bacteria divide, and that's how the colony grows. So if you let them grow over time moving from left to right, you get 4 cells, 8 cells, 16 cells. And each one of those bacteria have a genome. Now the question up here at the start of class was something about Meselson and Stahl, regardless of whether you got this out of their manuscript, because of course it was written a long time ago and it's all scientific and I don't even like to read this paper. Oops, I recorded that. and Stahl of Meselson and Stahl actually taught me biology when I was an undergraduate, so I hope he's not listening to this video. What did they grow the bacteria in initially? They grew it in a special type of media that had heavy nitrogen. N15. So normally nitrogen is 14. Look, we've got even the music classroom, we've got a periodic table over there. Right. N14. So they grew the bacteria initially in heavy N15. So I'm going to draw their genomes as blue circles, and when they're heavy, I'm going to draw them as thick blue circles. Okay, go figure. And because these are double-stranded, oh dear. Let's see how this works. I'm going to draw that as, maybe I'll make it a little bit smaller. So there's the 2 strands of, so imagine that's a double helix, 2 strands of heavy chromosome and growing in heavy nitrogen. This is the pulse in a pulse chase experiment. What you do in a pulse chase experiment is you pulse some sort of chemical or dye or tracer for a short period of time into, say, in this case, an organism. Then you take it away so that you know that all of the organisms or cellular vacuoles or components, or in this case, molecules, DNA, as time progresses, you know which ones were the original molecules that were labeled back during the short pulse of heavy nitrogen. And so what they have to do after they pulse is what? They chase. It's like, I don't know, tequila and beer, or who knows, right? You do a pulse of something that's heavy, and then you chase it with something light. Like water, I don't know. So we've got those heavy molecules, and now we're going to chase. They move into N14, which I'm just going to draw as black. So once these bacteria start to grow out, just after a few rounds of cell division, say, the second generation, they move the bacteria into unlabeled medium so that all of the new DNA that's synthesized has light nitrogen in it. So what happens in reality? So I'm not going to show you how this works for the hypothetical situation where conservative replication works. We're just going to walk through what actually happened in their experiment. Okay, so on the left, we've got in each of those cells, all of those cells are identical, so at some point, to save time, I'll probably give up drawing out all 4 cells, and then 8 and 16. I'll just settle on the one on the top. For this top molecule, what happens to those 2 chromosomes when the cell divides? The first thing we have to do, by the way, is DNA synthesis, so that that cell has 2 copies of the chromosome. So that's the same single cell, and then it divides. So what's going to happen? For each one of those genomes, in semi-conservative replication, the one strand of each heavy copy goes into both cells, which means that the light strand is the newly synthesized strand.
Yeah, if it's it's synthesized in
This is
chaser.
semi-conservative, right. So that second strand, the thin blue strand, is synthesized after the first generation of chase. So up until the part on the left, all the bacteria had been grown in heavy nitrogen, so their DNA contains only N15. After one generation following the chase, that is, you switch from N15 to N14. Ooh, man.
Shouldn't it be?
Don't read my handwriting.
Shouldn't it be on that intermediate stage?
Yeah.
One of them is also, the inner rings are lighter.
Right, so the inner rings during that replication stage, absolutely, we can make those lighter.
Lighter representing thinner?
Yes,
Representing the lighter nitrogen.
the N14,
Okay.
less
Less heavy.
heavy. So this is, on the left there, we'll call that generation 1. We'll call this generation 2 after the chase. And then we'll see what happens. Hopefully you'll tell me what happens in generation 3. So all we need to know to figure out what happens in generation 3 is how many original strands, not double helices, but how many original strands of the circular genomes exist in the third generation? The heavy ones, the thick lines. Is it 2 or 4?
4?
Yes. There should be 4 double strands from the original.
So 4 double strands go from the second generation where there are 1, 2, 3, 4 circles, right? There's a strand that's thin, a strand that's thick, a strand that's thin, a strand that's thick. Those are 4 strands that move into the third generation. And so those strands are either going to be, and we can assign these randomly to these 4 cells, they're either going to be thin or they're going to be thick. And in each of those cells, what would the other strand be, light or heavy?
They would all be light.
This is after we've removed the heavy nitrogen. All the DNA that gets created at this point is synthesized as light. So if we summarize what's happened, in that first generation, we can ignore all of this, the second, third, and fourth rows of cells. It's okay. Those would all look exactly the same as the top row we just drew out. It would just be copied and pasted. Same thing all the way down. Just synthesized, just trying to show you what happens as a colony grows over time. What percentage of the cells, there's only one cell, by the way, so do we have all heavy DNA, half heavy DNA, that is one strand is heavy and one strand is light, or all light? So column 1 over here. All of the cells in that generation are,
All heavy.
they're all heavy.
First generation.
First generation's all heavy. So we've got 100% in the first generation and 0 that have one thick strand and one thin strand, and 0 that have both thin strands or all light. So that was what Meselson and Stahl set up the experiment to do. They wanted to make sure that in the first generation, all of the bacteria had only heavy DNA, and then they wanted to watch what happened to those heavy molecules of DNA as time progressed. So what happens in what I've labeled here as generation 2?
Now you have 100% half heavy.
So each of these two molecules up here has one heavy and one light. So that's a half heavy molecule. So 100%, two cells that we're looking at in this case, but both of them have DNA that's made up of one original strand, one of the heavy strands, and one recently replicated or light strand. So we've gone from 100, 0, 0 to 0, 100, 0. And this is only for the case of semi-conservative replication. And I'll leave it up to you to figure out, if you want, to draw out what would this look like if conservative replication was happening? How would that differ from what we're seeing here? Let's finish up the third generation, though. But that's a good exercise to do, is to do exactly the same process, but assuming that conservative replication is true. So third generation, how does it change? Are there any all-heavy chromosomes in generation 3? How many are all light?
Half, 50%.
So we've got all light there and there.
There's 50% half.
And we've got the other 50% is half heavy. So in 2 generations, in the transition from the first to the second and the second to the third generation, you go from 100% of your chromosomes having only heavy nitrogen to 0 of your chromosomes still being all heavy. That happened in the first generation, actually. So this is what Meselson and Stahl expected to see with semi-conservative replication. So here we go finally to the bands. First question from the start of class. What do all these bands represent? Well, turns out nitrogen, when you call it heavy because it's N15 instead of N14, means it actually is heavier. So they took the DNA out of all of those bacteria. They took samples of the bacteria from every generation. They wanted to know how heavy is the DNA. We're not just talking about this academically. They actually wanted to know physically how heavy is the DNA. That's how they did this experiment. So you do something like, you take a tube, you fill it with a liquid, you add the double helices, and then you spin it in a centrifuge with centrifugal force pointing down, so that what's going to happen as DNA is being pushed, spun in a centrifuge, the heavier DNA's N15 containing DNA is going to do what? It's going to go farther faster. So you're going to get heavy DNA at the bottom of the tube. You're going to get all light molecules near the top of the tube, and what's in between, which I should have left myself space for, would be half heavy, half light. So that's exactly what you're seeing in this graph, this chart. You've got
Using DNA, wouldn't it? I mean, would you be denaturing it by spinning it at high speeds, or are the strands still intact?
Turns out the strands are still intact. So you're looking at whole chromosomes, and the heavier ones are farther down. And the band up here, it represents all light molecules. And in the middle, we've got anything that's half heavy. If I was magical and had better technology, I would be able to flip back and forth between the last slide and this slide. But you've got the model, so you should be able to compare the drawing that we just made to this chart. These are the actual data. What did we predict with semi-conservative replication? What did we start with? Generation 0, time point 0 in this case. Everything was heavy because they had done the experiment that way. They had grown the bacteria for generations in heavy nitrogen. Is that what they see here? Generation 0. Right? All of the DNA molecules are down here towards the bottom of the tube, all heavy. Okay. What happens in generation 1? What did we predict? All of those heavy, totally heavy molecules denature, and each of those heavy strands serves as the template for creating the synthesized or light strand. So everything at the first generation should be half heavy. And when we look at generation 1, right, there is no all heavy band, there's no all light band. All there is one big whopping half heavy band. Right? I don't need to beat the dead horse. What happens at generation 2?
Half of the half heavy.
Well, they didn't quite get to generation 2, they got to generation 1.9, but that's pretty close. They're equal ratios, as we predicted, 50% half heavy, 50% light. What happens when we hit infinity generations? If they did this experiment forever,
You would still have the original heavy strands, but they would be mostly light.
so isn't that interesting? What happens to, what does it look like is happening to these all heavy strands? They're dark, lighter, lighter. What's happening? How are they disappearing?
They're combining with lighter strands.
They're combining with lighter strands. So your original 2 double-stranded molecules that are all heavy get separated in that first cell division to be templates for creating half-heavy circular genomes. So they're basically being diluted by new DNA in the first synthesis and more new DNA in the second generation and more new DNA in the third generation. But those molecules are still there, we just can't see them. Which means maybe, which is kind of a crazy thought to me, but I don't know if it's true or not, somewhere inside me might be the original chromosomes that made me. Like one cell embryo, set of chromosomes from mom, set of chromosomes from dad. Like those original DNA molecules might be in my toe, I don't know. But those original molecules could still be, they're still in that experiment. You just can't see them because the newly synthesized DNA is all light. So as time progresses, the heavy disappears, the half heavy disappears, and you get this whopping overabundance of newly synthesized DNA that's all light. And again, I'll leave it to you to compare this to what you think would happen in conservative replication. Remember, that's not separating the 2 heavy strands and using them as templates. That's, you get it back. The all heavy molecules you get back after the first replication, and how that would differ from what Meselson and Stahl actually saw.
If it's conservative, then it would be half in the first generation, and then it would be light, so it would be heavy, light, but it would be half and half at all. Does that make sense? Like,
You wouldn't have any middle bands, you'd only
Yeah.
have,
You'd never have, exactly. So take home point, even if you don't want to draw it out, if conservative replication was happening, you'd have an all heavy molecule and then you would get back in the next generation an all-heavy molecule, and an all-light molecule. So you'd never, ever, ever see half-heavy. The only reason we see half-heavy molecules is because one strand is being used as the template to synthesize the other strand. That's why we see things that look like that. The old meets the new. Which has some interesting implications for construction of genetics and also the actual details of genetics. I think we've covered a lot of the details. I'm going to decide whether or not next class, I'm not done yet. I know 4 minutes left. Promise I'll get you out of here. We might go through a little bit more detail next class, but I want to get you to this exercise. So I'm going to leave this for you to decide whether or not you want to do it. This will not be graded. But creating a model for how this process works. So I'm going to distribute this as an exercise to you as well. Do it if you want to practice. It's coming through Google Classroom. And you'll see what the instructions are when you get the copy of this PDF. So I'm going to leave it at that. If you have questions, we can talk about that exercise next time. So to wrap up, we have a few things to do for next class. There is, I swear, one more survey for points. And then here's the information to focus on when you read that case study. And I've also developed practice for you, not for points. This is also coming, the sequence alignment is also coming to you via Google Classroom. So nothing that I've done today other than the first thing we all did together, that original Google Sheets alignment, none of the rest of it's due for points. It's all for practice. We can talk about answers next class if you want, but that's the one thing to focus on. Now I'm going to launch a Socrative quiz, wrapping up the day. This is also going to be scored. So you've got basically 2 points today. One for the Google Sheet exercise we did earlier, one for this right now. Yep. So all of the links are on Blackboard. Make sure you've done all 4 by next class. 5? Do all 5 by next Monday. You'll be fine. Somebody remind me that I told you that. Here, I'll write it down. Now, so we've got sequence alignment. That's been sent to you. We're all good. So as usual, I'm going to leave this going for some while because I asked you a couple of questions that are free response. So you don't have to complete it right now. Do it by 5 p.m., you'll be fine.